Here is the volatile ecosystem of the oral cavity. Rapidly dividing bacterial colonies sit in constant, direct contact with highly vascularized mucosal tissue. This space has a contradictory biological mandate. It must serve as a durable mastication zone, capable of crushing dense organic matter, while simultaneously maintaining a permeable mucous membrane. To manage the bacterial load in this environment, clinical dentistry has heavily relied on broad-spectrum interventions, specifically oral antibiotics and high-alcohol bactericidal mouthwashes. These methods operate on a principle of total eradication. They chemically strip the environment, destroying pathogenic microbes and naturally occurring beneficial flora with equal prejudice. The resulting sterile environment is inherently unstable. Without the beneficial bacteria that normally occupy the tissue surface, the compromised microbiome is exposed to aggressive repopulation, leading to chronic inflammation and secondary infections. To escape this cycle of eradication and reinfection, an intervention must be highly precise. It must differentiate between rigid dental enamel, soft vascular tissue, and microscopic pathogens. Achieving oral health requires precise biochemical management and the selective pruning of the microbiome, rather than the broad, sterile eradication of the entire environment. This diagram illustrates orthogonal photobiomodulation, using isolated wavelengths of light to trigger divergent biological processes simultaneously. The shorter 460 nanometer photons interact strictly with surface bacteria, while the longer 630 and 850 nanometer waves pass through rigid enamel to reach the soft tissue below. The BioLight Guardian Plus device is an engineered case study designed around delivering these exact therapeutic wavelengths. The hardware separates these functions spatially, featuring an upper row of blue LEDs and a lower row emitting red and near-infrared light. This physical separation ensures that specific anatomical zones of the mouth receive the exact type of phototherapy required for that specific tissue. Pairing these light arrays with an applied oxidative chemical catalyst creates a layered biological intervention that addresses both microbial load and tissue health. This targeted, multi-wavelength application allows for the precise, cellular-level management of oral tissues. We begin with the physical parameters of the upper LED array. It delivers 460 nanometer blue light at a strict irradiance, or power density, of exactly 2.2 milliwatts per square centimeter. Driven at this specific wavelength and power density, blue light functions as a targeted bactericide against harmful oral pathogens. This initiates selective lysing, the physical rupturing of specific cell walls. The 460 nanometer light excites porphyrins inside dark pathogenic bacteria, causing them to break down structurally, while light-colored beneficial bacteria remain stable. By surgically targeting only harmful strains, the mouth's biological scaffolding remains intact. Leaving these beneficial strains unscathed allows them to multiply. They naturally outcompete returning pathogens for surface area and nutrients, proactively defending the enamel. The 460 nanometer wavelength also serves a secondary function. It accelerates specific chemical reactions. When paired with specific whitening compounds applied to the teeth, the blue photons carry enough energy to act as a catalyst, speeding up the breakdown of deep enamel stains. Blue light therapy utilizes the physics of the microbiome to the patient's advantage, functioning as a microscopic scalpel to dismantle pathogens and accelerate targeted chemistry. The Guardian protocol pairs blue light with a specific chemical catalyst, carbamide peroxide, formulated at a concentration of 36. Controlling the kinetic release profile of the oxidizing agent allows the light to accelerate the reaction on the enamel while protecting delicate, vascularized tissue. The bottom row of the device handles a different biological task, outputting 630 nanometer red light and 850 nanometer near-infrared light. This lower array is calibrated to a specific irradiance of 1.2 milliwatts per square centimeter. These near-infrared photons bypass the rigid enamel entirely, penetrating deeply into the highly vascularized periodontal tissue below the gum line. The 630 and 850 nanometer wavelengths perfectly align with the absorption spectrum of specific chromophores, light-absorbing molecules, housed within human mitochondria. Inside the mitochondrion is the primary target for these photons, an enzyme called cyanchrome C oxidase, a critical component of cellular respiration. Striking this enzyme with targeted light forces the mitochondria to increase their production of ATP, the base molecule responsible for cellular energy. By increasing the energy reserves of these cells, the localized tissue can modulate inflammation and increase local blood circulation. Clinically, this bioenergetic cascade directly accelerates periodontal wound healing, reduces gingivitis, and actively soothes oral mucositis. Red and near-infrared light increase the bioenergetic capacity of the gums, equipping the cells to actively repair mucosal tissue. These clinical outcomes depend on strict dosimetry, the physical delivery of the exact correct amount of light to the exact correct location. This schematic illustrates how the Guardian Plus mouthpiece solves the problem of spatial distribution. Using specialized optics, each diode projects a 120-degree beam angle, overlapping to ensure gapless coverage across the entire dental arch. To prevent under- or overdosing the tissue, the system relies on an automated, standardized 5-minute exposure cycle for every session. Because the device houses blue LEDs strictly on the top and red and near-infrared on the bottom, patients require a specific protocol to treat both arches of the mouth. To achieve uniform exposure, the patient conducts a 5-minute session, rotates the device exactly 180 degrees, and initiates a second 5-minute cycle. This mechanical rotation ensures that the microbial pruning of the blue light and the deep mitochondrial stimulation of the red light are evenly applied to the entire oral cavity. Integrating precision phototherapy and stabilized chemistry establishes a calibrated biological protocol for maintaining oral tissue and microbial balance.